Hello all, we're going to be talking about genetics part 4, karyotypes, sex linked, and pedigrees. Now what is a karyotype? A karyotype is a picture of all of the chromosomes. Now typically we take a picture of them and then we maneuver them around and line them up into pairs. So we see chromosome number 1. Now they may not be exactly in the correct shape, just like this one is kind of curved in, but all of the bands match. And this is chromosome number 1 and our second chromosome number 1. And we see chromosome pairings 1 through 22, which are going to be our autosomes or our body chromosomes. And then we have our 23rd sex chromosome. In this image, we see an X and a Y. Now this is the only pair that is going to be different if you are a male. Now if you're a female, you will have 2 X's, which will mean they will look the same. So again, the sex chromosomes, which is going to be the 23rd pair. If you are a male, you have an X and a Y, and if you are a female, you have an X and an X. Again, chromosomes 1 through 22 are your autosomes, or your body chromosomes. Now remember, when a woman makes her eggs, she has 2 X's, so the only thing that she can put into an egg is 1 X. So every egg carries a single X. However, males make sperm and they can either put in an X or a Y. So half of the sperm carry X's and half of the sperm carry Y's. So gentlemen, you are the one that actually determines the sex of the baby. So when you get a girl and you really want a boy, you blame yourself. Sex-linked traits are characters or traits that are found on the sex chromosomes. They're linked to those sex chromosomes, that 23rd pair, that X and that Y. Typically, it's either on the X or linked to the Y, not both. Y-linked disorders are going to be found on the Y chromosome and only affect males, because only males carry a Y. For X-linked disorders, they are found on an X and can affect both sexes, both males and females, because both male and females do have an X chromosome. Now for females, they must have both X's affected, so both of them must have the mistake or the disorder to actually show that disorder. Now that's good for the females, because if one has the disorder and one is normal, this normal one will take over and the disorder, or the one that has the mistake, will disappear. Now for the gentlemen, they only have one chance, one option, because they only have one X. And if it's the one that has the mutation that causes the disorder, then they will show the disorder. Now when we talk about sex-linked in a Punnett square, we are going to talk about X's and Y. Now we do talk about dominant and recessive, talking about the alleles above the X and the Y chromosomes. So here we have the mother put up top, because the mom has an X and an X. And the mother in this case will be heterozygous, talking about color blindness. Color blindness will show if you have 2 little b's in females, or a male has 1 little b. Now for the father, which is an X and a Y, the Y does not have blindness or color blindness on it, so we just leave it as the Y. But the X, the male is normal, so he has a big B. Again, for a Punnett square, we're going to bring it down and across to get X big B, X big B. So this is a normal female. And then we have X big B, X little b. This female is also normal, but it is known as a carrier because it does carry one X that has the mutation on it. Now, looking at the gentlemen, or the males, we have X big B, Y, which is a normal male, but we also have X little b, Y. Now, again, in males, since we only have one X, they only have one try to get it right. And this X has the mutation. So this gentleman right here would be colorblind. So in the genotypes, we'll list out all of the genotypes. And the phenotypes will have 2 normal females. Remember, though, one is a carrier. And 1 normal male and 1 colorblind male. Now looking at hemophilia. Hemophilia is also X-linked. Hemophilia is recessive. So again, for the females, we must have 2 little h's. but for the males, we only have to have one little h. Now we have a heterozygous mother, big H little h, and a hemophilic father. So he does have X little h, so he has hemophilia. When you bring it down and across, we end up with X big H, X little h, 25%. X little h, 25%. X big H, 25%. Y, X little h, Y. So when we talk about the phenotypes, we're going to have one female that's normal, that's a carrier, one female that has hemophilia, one male that is normal, and one male that has hemophilia. So yes, in the phenotypes, you do need to break it down into males and females this time, not just normal or have the disorder. Now, pedigrees is another way to show how traits are inherited. In this pedigree, we have 3 generations. Each generation is distinguished by a Roman numeral off to the side. This is generation number 1, generation number 2, and generation number 3. Now, we do typically have a key below as well. Males are squares, females are circles. Affected with the disorder or the trait is going to be filled in, and deceased tends to have a line going through. Now, let's go ahead and take down the chart that goes through all the different aspects. Again, male square, female is round. Mating, they have a horizontal line connecting them. A parent, so they have the horizontal line, which means mating, and then you have a T, or a tree that comes off of them, and shows the children, or the offspring. Typically, the firstborn is listed first, then the second, but if they don't tell the ages, then don't worry about the order. Non-identical twins are shown at an angle instead of a square. Monozygotic twins, or identical twins, are going to make a full triangle. Unspecified sex would be a diamond shape. We don't know what it is. Number of children. Affected individual would be solid colored. For carriers, you can show them half and half. You don't have to, but a lot of times you'll see half and half, which means a carrier. Now, that doesn't always mean that they show the disorder. That just means that they are carriers, or they're heterozygous for the disorder. A dash is death. Abortion of a stillborn is a line with a solid dot. And a miscarriage is just a triangle. Now, if we look in the olden times, when we're looking back at the kings and queens and royal families, a lot of the times you see a double line. Well, that's consanguineous marriage. And that means marriage within the family, or incest. Now, using the pedigree, imagine that you are a genetic counselor and you have to figure out whether albinism is inherited as a dominant or recessive. And a couple has come to you and provided you with this tree, and it's your task to determine whether, again, albinism is dominant or recessive. Well, how do we do that? Well, first we're going to look at the pedigree. And then we're going to assume that albinism is dominant. And if it has a big A, then they will be albino, which means it has no pigment or color. So that means these 3 here that are affected, that have albinism, should be big A dash. Big A dash. Now again, we don't know what the other letter is, but as long as it's got one dominant, then it should show. So that means all these other ones that are not affected have to be recessive. Let's add some recessives in here. Little a, mating with little a. Wait a second. Can these 2 parents create a big A? No, they can't. They don't have a big A to give. So albinism must not be dominant. So let's check the recessive. Recessive. So if albinism is recessive, we'll go ahead and put the little a's in. Okay? That means all the other ones have to have a big A of some sort. Well, does that fit? So if we do that, okay, we can have that. We'll add in all the other A's. The question mark means that there's no way to tell what the other letter is either going to be capital or lowercase. So we add a question mark so that we always distinguish that there are 2 alleles. We just don't know what the second one is. Now, looking at this little a, we know that they have 2 little a's. So one of the parents had to have a little a and the other had to have a little a. So we do know the genotype for these 2 parents. Well, Same with this parent right here. This child has little a, so they got one from the dad, and it must have gotten one from the mom. Now, we know that one of these two parents had to give a little a. Now, it doesn't matter which one you give that little a to, as long as you delineate the other one with a question mark. And that's how you create a pedigree. The human genome is all of the genetic information. It is a complete set of the human genetics. Now, you've heard that the Human Genome Project has actually been successful. We've completed and figured out all of the genes of the human genome. Now, with this, we can actually see different mutations and different genetic disorders. And you can actually have your DNA sequenced and find out if you are at risk for different genetic diseases. Well, that's all I got.